Yeah. Okay.
Okay, well, shall we get started? It's actually a great pleasure to welcome Alanna Watt from McGill. She escaped the ice storm to come and join us here in sunny Hamilton. So Alanna actually did her undergrad at Concordia and then went to Brandeis for her PhD to work with Gina Torrigiano. Some of you the older, more grizzled folks in the audience may remember Gina coming to speak a few years back here. And then went to across the Atlantic to do a postdoc with Michael Heuser in UCL. Very interesting stuff in cerebellum. And so now she actually works in cerebellum and has chosen not to talk about all of her interesting work on the cerebellum's role in ataxia in a number of disorder models, but rather a developmental story on the connectivity of the output of the cerebellar cortex. So we're very pleased to have her here. And I'll just let folks know there's a reception afterwards if you wish to chat with her a little bit afterwards. So please come up to the second floor after the talk as well. Okay.
I'll turn that on. Can you hear me okay? Excellent. Okay, great. Thank you very much, Dita, for the invitation. I've had a really good day today, and I'm looking forward to the rest of the day as well. Having dinner with grad students, for instance, which sounds like it's going to be a lot of fun. And yeah, I'm actually so excited to tell you about this story. I'm going to tell you one story that's just, kind of new, and this is really one of the first times I'm talking about it to a larger audience, so it's very exciting. And then at the end, I'm going to tell you more of the developmental story, which is mostly already published, but I have some interesting things with that too, I hope. So, as alluded to, I'm really interested in the cerebellum, which I'm showing here. Oops, I need to turn this on. This is, cerebellum means little brain in Latin, and it is kind of tucked up below and behind the rest of the brain in humans and at the back of the brain in rodents. And it's a brain region that's been involved in many different things. It's most famous for its role in motor coordination, balance, and motor learning. It has non-motor cognitive functions, reward functions. There's all sorts of new things being constantly discovered about what the cerebellum is doing. And that makes it a really exciting thing to study. And I'm going to be telling you a little bit about what we are interested in the cerebellum in particular. And so what we study in my lab, we have kind of two different streams. We have a basic science stream where we really just are wondering how the cerebellum is functioning, how it's developing. And we're looking actually now, I really came, my history, my background is with developmental neuroscience, but I've really transitioned, and sometimes we also even look at aging these days. So we look across an animal's lifespan. And then we also have some applied science questions, like what happens when the cerebellum is altered in disease? And we look at two forms of ataxia, spinocerebellar ataxia type 6, and then a form of ataxia called RSAX, or ataxia of the Charlevoix-Saguenay region. And this is actually an ataxia that is more prevalent in northern Quebec, so we can think of it as a Canadian disorder. And I'm not going to talk about this, but I'm happy to talk about it with anyone who's interested, because I wanted to more focus on some questions that I thought would be interesting for Dita and other members of the Gillespie Lab here. So I'm going to tell you a little bit about the cerebellar microcircuit. So this is a slice of a mouse cerebellum, and you can see that it's pretty structured, right? So this is a cartoon from a review paper I wrote many years ago now. And you don't have to kind of know what everything's called. What I just want you to see is that there's just a few different types of cells, and that they're color-coded here. So there's like some blue cells, some orange cells, etc. But there's not like dozens and dozens of cells. It's really just a handful of cells. And they're organized in a very regular way. So these orange Purkinje cells, those are the one cell type that I am going to be talking about a fair amount. They're kind of lined up here. All their somas are in a single cell thick layer. And then you've got these granule cells down here. Those are the blue ones. It's very structured. So just like you see in this slice of a real cerebellum, it really is very, very organized. And so people have been very drawn to the cerebellum because they like this interesting structure. And people have looked at it in a number of different ways. Okay, so as I said, Purkinje cells have kind of been the star of the show for a long time in the cerebellum. First of all, they have these amazing dendritic trees up here. They're just really big. They receive hundreds of thousands of synaptic inputs. So they've been studied from their interesting role in synaptic integration. And then their axons project out of the cerebellar cortex. And in fact, they're where these cells are really important for conveying anything that's happening in the cerebellar cortex out. People have often thought that these are then the output cells of the cerebellum, but that's not true. They actually make a stop along the way out, and the Purkinje cells make synapses onto neurons in the cerebellar nuclei, or the CN. Sometimes people call these the deep cerebellar nuclei, or the DCN, but there's kind of been a shift, and now we call them the cerebellar nuclei. So that's what I mean with the CN. So let me just show you again in a little cartoon form. So here's the cerebellum. You've got this big cortex where the Purkinje cells are all lined up, and they're all projecting to these cells in the cerebellar nuclei. So information, this is what these cerebellar nuclei cells look like. They look kind of like this, you know, just sort of multipolar cells. So information comes in through the cerebellum, to the cerebellum. It gets processed in the cerebellar cortex. And, cerebellar cortex information processing happens here, and then the information goes to the cerebellar nuclei, and then those cells project to other structures, including like the VTA, the hippocampus, the thalamus, brainstem, and spinal cord. So there's a lot of information going from the cerebellum to extracerebellar locations, like the rest of the brain. So these are the cells. These CN neurons are really the cells that project out. And there's been a lot of interest in understanding where they project, what they actually do. One other thing I need to tell you about, so you can understand why we're asking the question that we are, is if we take a slice through the cerebellum, and you can see that it's organized in this like folded, it's a very folded structure, and it's very characteristically folded. These different areas are associated with different function. So first of all, you can see that there are these lobules, and they're numbered. The lobules of a mouse look a lot like the lobules of a person or of a bird. And those lobules have been organized into functional zones. And those functional zones have really catchy names like anterior zone and posterior zone, you know, really great. But what we know is that they are associated with different functions. So anterior is sensory motor. Remember, this is very much a motor area. But there's also some parts of the posterior where there's thought to be cognitive and emotional processing. So very different types of information being encoded there. Okay, so the question we set out to ask was, how are Purkinje cells organizing their projections onto these cerebellar nuclei neurons, or CN neurons? So we know people have estimated that there's maybe 40 Purkinje cells converging per CN neuron, but we haven't really understood where they're coming from. Which Purkinje cells are converging onto which CN neurons? You might think, well, should I really care about this? And I'm going to try to show you that yes, you should care, because it's going to really change what these CN neurons are encoding depending on how this convergence is happening. So imagine that we have convergence from Purkinje cells within the same zone that are kind of encoding the same kind of information. Well, then that CN neuron is going to be encoding something kind of similar, right? But imagine that we have convergence from Purkinje cells that are actually coming from across zones. So they're actually encoding different kinds of information, and that information is now converging on the same CN neuron. Well, what that CN neuron is going to be conveying to its downstream targets is going to be very different. We didn't know which of these kinds of encoding was happening in the cerebellum. So what I'm going to be telling you about is work that's been done by a graduate student who's soon to be graduating, Kim Groover. And she's really been phenomenal, because I'm going to be telling you about some pretty challenging experiments that have been really tough to do, but she has really risen to the occasion, and she has done some amazing amount of beautiful work, as I'm sure you're going to see. So we're using transgenic mice that express EYFP with channel rhodopsin in Purkinje cells. So you can see that the Purkinje cells are lit up. This is actually their axons being lit up. With the EYFP, they're also expressing channel rhodopsin. So they're going to be light sensitive. And we use a spatial illuminator that allows us to focally stimulate really, really precisely. And I'm going to show you that in a second. So if we make a tiny little focal box of, say, 15 by 15 microns, and we put that over a Purkinje cell soma, and we're recording from the Purkinje cell, well, it's a we're going to activate these light-activated channels, and we're going to be able to cause action potentials just as you expect with channel rhodopsin. If, however, we move that box into the molecular layer, into the white matter, and we don't actually hit the axon, well, we're able to focally stimulate very, very carefully, and like the light isn't kind of going everywhere. You can see that we can stimulate and not see a response at all, or we can move over the stimulation 10 microns, and now we can stimulate and see we can get action potentials when we activate the axon. So this is showing you that we can activate Purkinje cell axons with light and make them spike, right? Make them have an action potential. So that gives us a tool for probing connectivity between Purkinje cells and their downstream targets. Here, of course, we're just recording from the Purkinje cell, just sort of proof of concept. Okay, so now what we're going to do is try to see what kind of encoding is actually happening with the Purkinje cells and the cells of the cerebellar nuclei. So now we're recording from the CN, from these cerebellar nuclei neurons. And these are really tough cells to record from, but Kim is an expert at it. And then what she's going to do while making this recording is she moves the stimulation around the cerebellar cortex. So you'll see what I mean as I show you. So first she's just stimulating this bundle of axons. No response, right? So here's the light and here's the trace. So that doesn't seem to do anything. There isn't a synapse coming from this region being made onto this cell, the one cell she's recording here. But she moves and looks in another location, over at this lobule 3. No response. Then she looks over here, lobule 4-5, again, no response. Then over here, when she stimulates in lobule 6-7, all of a sudden, I hope you can see there's a nice big response, a big synaptic response between Purkinje cells in this lobule and this cell. She goes on and does other, and in fact is able to systematically activate Purkinje cells in each lobule and map out where are those inputs coming from. And in here, I think this cell is pretty clear. So I'm going to show you back to that idea we had about the different kinds of convergence we might see. Well, what I just showed you, it's going to be an example of this, right? Convergence between neighboring Purkinje cells. So we have all the different lobules here, now just lined up from 2 to 10. And you can see we see a nice big response in lobule 6, 7. That's the same cell I showed you. I'm just rearranged the traces so it's a little bit tidier. Okay, so that's an example of convergence between neighboring Purkinje cells. This is likely, it's actually a pretty big response, so it's likely more than one Purkinje cell. Sometimes Kim saw something different, though. Sometimes she would see more than one location would elicit a postsynaptic response, like here, where we see a response elicited when she stimulates lobule 4, 5, and also when she stimulates lobule 8. So here now, we have two different lobules converging on the same postsynaptic cell. And here's another example where she saw nothing in lobule 2, 3, or 4, 5, but then a big response in lobule 6, 7, another response in lobule 8, and even a response in lobule 10. So now we're seeing different kinds of patterns of inputs onto different cells in the CN. Okay, so then how do we make sense of this? Kim did this a lot. These are challenging experiments, but she was able to build up a map, sort of maps of what kind of input we tended to see. So first, I just wanted to tell you that we don't, even though you can see in some cases there are big inputs, in some cases there are little inputs, there's nothing systematically different between the amplitudes coming from different lobules. So you see the same kind of spread of data. So it doesn't seem like there's one input preferentially like dominating the others, one lobule dominating the others. It's kind of random. We see a lot of different patterns. Okay, so I've talked to you about these different functional zones. Well, is there anything interesting about that in our data? So now I'm showing you all the data that Kim collected where each cell is a line. And so what she's done is mapped out where the inputs are coming from, which Purkinje cell lobules are converging on a particular cell. And so here you can see there's a lot of cells, a lot of input. Down here, this is a cell that's only getting input from lobule 6, 7. And then we did a clustering algorithm to this data. And what's interesting is that this is the cluster. So this is clustered on her data. And what I hope you can see is that what comes out of these clusters is actually these functional zones. So the central zone, and then you've got the anterior zone here, the nodular zone, this lobule 10, and then the posterior zone. The inputs are actually clustering. So these functional zones that are defined from like the inputs to the Purkinje cells are still visible when you look at the output of the cerebellar cortex. So we see that this zonal structure is important, and it appears to be maintained at the Purkinje cell to CN synapse. Okay, so that's pretty cool, but probably what you've seen already in this data is that we don't just see these zones, right? We have cells where we see a lot of input, not just single zones. So if we just look at sort of which cells receive one zone input and which cells receive multiple zone inputs, lots of cells receive multiple zone inputs. So what can we say about that? Well, to look at that, we built a model. So this was a model looking at connectivity where we assumed that the connectivity would be random. And so what we're looking at here is kind of what we saw with our connectivity, what we're calling the actual connectivity versus a predicted occurrence if this connectivity is just random, okay? So you could imagine then that you're just going to sometimes get connectivity from one lobule, sometimes from another. more infrequently, you might get inputs from 2 lobules, and even more infrequently, inputs from 3 lobules and 4 lobules, etc. And so what we see here is that for most of the connectivity, it's around or close to 1, which means that it's just what we would predict randomly. But occasionally, we have these big differences. And those big differences are seen for input that's coming from multiple lobules. So we actually think that these cells that are getting 3 or especially 4-zone connectivity are kind of special because we're seeing them much more frequently than we would expect to. They're overrepresented in our data. You would expect to see them very, very infrequently, but we see them a lot more than we expect. So 4-zones cells seem to be kind of special. And if you think about what that means from this kind of encoding perspective, we have 4-zone cells that are going to be encoding different types of input. These CN cells are getting input from all those different types of Purkinje cells. Okay, so we see 4-zone connection motifs with kind of input from all these different functional zones. That's pretty cool, and we were very excited by this data. I can tell you that, it's not exactly what people had reported in the literature. It was not exactly what people had predicted. So I think a lot of times when people see nuclei like this, the idea is that there's not a lot happening as far as convergence. It's pretty simple. Sometimes people talk about relay neurons. There's not much going on there. All the kind of interesting computation would be happening in the cerebellar cortex. We thought, well, okay, our data, functional data showing synaptic connections is pretty cool, but anatomically, people haven't seen this kind of evidence. So we decided to try to look ourselves using viral expression of viruses, just expressing 3 different fluorophores. So we have a TD tomato, a GFP, and a cerulean. So just a kind of red, green, and blue, which we injected. And this work was done by Evyatar Fields, another graduate student. And so he just injected into different lobules. And this is showing you an actual brain that he was able to inject with 3 different fluorophores. So anterior is here. Some of these cells are labeled purple. Some of the central cells are labeled with green, a little bit of the nodule area as well. And then the posterior is labeled with blue. So basically, we're just trying to hit different lobules with different colors, different functional zones with different colors. And then we want to look into the actual nucleus and see what the projection patterns look like. And so remember, we're not labeling every single Purkinje cell within these structures. It's just sort of a sample. And what you can see is that actually, there are differences in the projection patterns, right? Like the purple doesn't look exactly like the green or the blue. They do have some kind of gross morphological or macro scale differences in the projection patterns. And when you superimpose all 3, you can see that there are some differences. However, if we look really closely at the puncta, so the presumed synaptic sites, and we just randomly choose different areas. And actually what I'm showing you, when I show you some data, it's going to be for all the areas, but I'm just sort of zooming in. And so you can see there's puncta here that's really, close to other. So we have purple puncta here, there's a blue puncta over here, and some green puncta. They're all pretty close to each other. This is 20 microns apart. Over here, a different location, we see similar things, right? So we're not saying that all the projections are all intermingled, but when we look across this nucleus, we actually see that any one color is pretty close to another color. And if we look at the distance to the nearest neighbor of a different color, this is what I'm showing you here. And it's just shown in logarithmic to kind of make it a little easier to see. So this is microns. And basically, we're going from 1 to 128 microns. And that's how close one puncta is to another puncta. So you can see up here, we've got almost 100% of all the puncta are within 128 microns of another color. okay? And so what does that tell us? Well, I mean, you can see there's a wide variety of distances from each other. We're definitely not hitting all of them, so this is exactly what we would have expected. But from a biological point of view, what does this mean? Well, if we think about what is the average size of a CN neuron soma, that's shown here. So it's about 17 microns. So that means that, you know, almost 30% of all the puncta are within the size of another soma to each other. So that's certainly close enough to be making synapses onto the same cell. And if we look at the average dendrite length, well, basically more than 80% of the puncta that we see are close enough to be making a synapse with another cell. Okay. So we feel this is strong anatomical evidence that supports the idea of multi-zonal convergence on these CN neurons. So even though at the macro scale the projections look a little different from each other, when you really look down at the puncta, they are close to each other. And that supports the electrophysiology, the functional data that we have. Okay, so is there a spatial organization to the different cell types? So what I'm showing you here, it's a little hard to see. I think it's a little bit bleached out. I'm sorry. So Kim is a very careful scientist. Every time she records from a cell, she takes very close notes and takes pictures of where she's located within the nucleus. So then she's able to go back and recreate kind of like where each cell lies within the nucleus. That's what this is showing you here. These are the lobules, and here's the actual nucleus. And then this is each cell with each kind of projection pattern. So we have cells that are just getting a central zone input, cells that are getting posterior and nodular, and you can see that they're just scattered all over. But if we just look at the number of zones, that's what I'm showing you here, there's, you know, it's a little bit hard to see. Is there maybe some organization with that? Well, we thought that there might be. So you can see the 1 zones are in white. The 2 zones, it's sort of going from 1 to 4 in different levels of gray. So the 4 zones are dark black. Okay, so what might it look like if there was some sort of spatial organization? So one thing to know is that in this structure, there's been some what we're calling this axis here, the RV-CD axis, is an important axis, and there is some organization along this nucleus in this axis. And so we wondered whether there was any kind of organization in the cells that we were recording from. So we reasoned this way. If there wasn't any topographic organization, and we just like kind of plotted their coordinates, and then maybe, you know, looked for a correlation, we would expect there to not really be any correlation. There wasn't any organization. But if there was, we might see that shown with something like this, that there's some sort of almost stripe in the data, and we would see a higher R-squared value, a higher correlation between the data. There seems to be some sort of relationship between these cells and their location. They seem to be topographically organized in the nucleus. Okay, so what does it look like for the different cells? So if I'm just separating them out now, we have cells that didn't have any responsive lobules. I didn't talk about these cells yet, but sometimes when Kim recorded cells, she would record and not get any stimulation, but if she stimulated right over the cell itself, there would be a response showing her she had made a good recording. It's just that it wasn't getting any input from the cells there. And then we have 1, 2, 3, and 4 zone cells. Okay. So if we look at the correlations, well, these two are pretty low, but then they get pretty high over here on the 2, 3, and 4 zone cells. So is that meaningful? It's kind of hard to say because we have also smaller numbers of inputs, right? So it's easier to get higher correlation when you have smaller numbers of cells. So to look to see whether this was kind of like just something that looked correlated or whether it really could be correlated, we did something called bootstrap analysis. So bootstrap analysis is like this. We have all this data, and we're just going to randomly sample the data we collected and see how likely is it that we see something. So what I'm going to be telling you about is a p-value, and the p-value isn't the fit of the correlation, the p-value is really about the distribution of the correlation. So here, let's imagine we're taking 10 cells, and then we randomly, so we have 10 cells. So let's just randomly sample our own data, each time picking 10 new cells and looking at the correlation. So here we see something like this with a low correlation. Here we pick another 10 and we see a high correlation. And we do that 50,000 times. So many, 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 many times. And we're able to build up a profile of like what the distribution of R squared values could be, just with given the constraints of our data and the shape of the nucleus and things like that. And this is what we look at, what it looks like. And then we just say anything in the top 5% is going to be a p of 0.05 or less. And so if anything is up there, well, maybe it is actually going to be more significant than we'd expect randomly. So when we do this, the only p-value that's significant is the 4-zone cells. Of course, this is also the cells that have we have the fewest of these cells, so you have to take it with a grain of salt, but it is statistically significant. We do see a difference. We do see a p-value of less than 0.005. And if we look along the medial axis, we can do the same kind of thing. We, again, don't see any significant difference in this case. So that kind of suggests to us that, we've done this now many, many times, and the only kind of significance we see is for these 4-zone cells. So we think that that suggests that these inputs are possibly topographically organized. These cells are topographically organized within the nucleus. They seem to be lying on this particular axis. Okay, so I'm hoping that you guys are thinking, wow, this is kind of interesting. We've got these 4-zone cells that seem to be, maybe localized in a special way, and they're perhaps getting input, these cells are getting input from really different kinds of, Purkinje cells encoding different kinds of information. So how might that function, right? Like how might that be, do those cells have to work together to make the CN neuron fire, or are they working independently of each other? So these are tough questions to answer. But basically what we wanted to ask was, how are our inputs affecting the output of the CN neurons? And I need to tell you a few things so that you can understand why this is a hard question, okay? So first, I'm going to tell you about the experiment that Kim did to try to address this. And this was a very, very hard experiment. Like, this was really tough, and you'll see why, I think, in a second. So I'm not sure how many of you are electrophysiologists. If some of you are a whole cell electrophysiologist, you know this already, but if you aren't, let me explain to you how it works. When you go to make a recording from a cell, the first thing that you do is get a seal, a really nice seal onto that cell. And that's what Kim is doing here. That's what I'm trying to illustrate here. And then you break through, and then you can assay the synaptic milieu. You can like listen to what's going on at the synaptic level. Here, when you have a seal onto that cell, you won't be able to see the synapses, but what you can see is action potentials, spikes, because they're really big. So it might look something like this, because one thing that I need to tell you is that these CN neurons fire action potentials spontaneously. So that means they don't need any synaptic input to make them fire an action potential. They fire action potentials like clocks. They have this intrinsic activity. Okay? So that's interesting. So then we have to think not in terms of like a synapse making a cell spike or not. We're thinking about how is a synapse influencing a cell that's already spiking regularly. The other thing I need to tell you, which probably some of you know, but some of you might forget or not know, is that Purkinje cells are GABAergic. So they make an inhibitory synaptic connection onto these cells. They're releasing GABA. So we need to think in terms of an inhibitory cell and how a synapse, and how that synapse is going to affect a spontaneously active firing cell, right? So It's not going to necessarily be driving it to spike or not spike. It will probably be more modulating the timing of its action potentials. Okay, so with that in mind, what I can tell you is that Kim did these experiments where before she did a whole cell recording, she tried to just get a cell-attached recording, and then once she had a nice cell-attached recording and she could see the spiking in the cell, she then stimulated in all these locations where we were stimulating the lobules. So just like what she did earlier, and then she recorded action potentials, right? And then what we looked at was the timing of the duration of the action potentials just after the light pulse. So this one is 1.46 milliseconds, 1.54 milliseconds. Over here we have 2.41 milliseconds. It's all around 1.4 to 1.6, other than this one exception that's a little bit longer, right? 2.4 milliseconds. So is that interesting? Is it important? It's hard to know, right? So what's so cool about this experiment and why this experiment was so hard is that after she did this, then she broke through and recorded from the synaptic response. So she could see the spiking output of the cell and then the synaptic input of the cell. Unfortunately, in that order too. So lots of times when she did these experiments, she wouldn't be able to break through. It's a really, really tough experiment to do, actually. I'm really impressed that she could do it. So let's see now. So now she did the same thing, stimulating in these different locations. And see this. The only time she saw a synaptic response was over here in this lobule 6-7, where we saw a pause that was longer, right, than the others. So now it kind of tells us something, right? This synapse seems to be causing a pause in the spontaneous activity of the cell, which is what you would predict when you have an inhibitory synapse being made onto a spontaneously active cell. The synapse isn't big enough to turn the cell off entirely, but it's just enough to kind of delay that action potential a little bit. So there's a little bit longer pause than we would expect otherwise. Okay, so how do we actually analyze this kind of data? It's obvious, it's not totally obvious, but what we did was we measured something called the ISI ratio, where we looked at the ISI, so the spike interval after the light pulse, and then we looked at the average of all the other ISI durations. So in this case, the ISI ratio would be a little bit longer, so 1.5. And you might be thinking, why not just measure it as milliseconds or something like that? But what I haven't told you, and it makes it a whole lot more complicated, is that each cell fires at different frequencies. So one cell might be firing at 20 hertz, another cell might be firing at 40 hertz, and of course then the pausing and the duration of that will be different. So we kind of need to look at it in light of the action potentials that it's firing at baseline. Okay, and then this I have to say is Kim's favorite graph from her entire PhD. And if you look at it, you can say, yeah, I can understand why. Look at that beautiful R squared value and P value. So what she has shown here is if we look at this ISI ratio for this 4 cells where she's been able to do this experiment, because it's such a hard experiment, where she was able to look at the pausing, that's shown as the ISI ratio, and then look at the amplitude of the synaptic response once she breaks through, we see this really nice linear relationship. And what made me very interested with this is that what you can see, and it might be a little bit hard to appreciate this, but these are pretty small, right? Like, so the largest one we see is about 100 picoamps, and we're down to like under 20 picoamps. And most of them are big enough to cause a spike. What this gray here is showing you is like the variability of the background spiking. So in this case, the very smallest one seems to be within the range of the variability in the background spiking. But most of them, even small inputs of about 60 picoamps, appear perfectly adequate to shift the spiking on its own. And so that's actually really interesting and important because people have thought that it would take much larger synaptic inputs to be able to alter the output. So small inputs like 60 picoamps, that's like more than half of all the inputs we measured. So it really shows you that they actually are probably kind of powerful enough to affect the output even without needing to work together. So what we think that this means, and it's really just one of those things like it's a maybe, right? Like the inputs from the different zones may work independently. It doesn't appear that they have to work together. They don't have to kind of all sum up to be able to change the timing of the spikes. Even little inputs can change the timing of the spikes. And ultimately, from a point of view of neural coding, that's what's important. They may still work together. We don't know that they're not doing that, but at least our data shows that it doesn't appear that they have to work independently to do so. So that's what I hope you can take from that, that data. Okay, so this is part one, and I'm going to summarize it really quickly. So what I've shown you is that we see both single cell and multi-zone inputs to individual CN neurons. So the kind of projections that we see from Purkinje cells to CN neurons are quite varied, right? Like we don't see just one particular type of cell. We see a whole range of cells. But we think that these 4-zone cells are more prevalent than we would expect. So they seem to be kind of special within the circuit. And we also think that they appear topographically localized. As I said, take that with a pinch of salt. We don't have a huge N, but we do think that it looks, based on our bootstrap analysis, it does look like there's something there. And then finally, what we showed you is that even small inputs on their own seem capable of altering the output of the cell. So they seem to be able, this at least suggests that these different inputs could work independently and they don't have to be kind of synchronously active to have an effect on neural coding. Now, they may do so anyhow. We can't really conclude one way or the other, but at least it suggests that the way that these synapses are encoding information could be independently, so that we have the different zones not necessarily working together, but actually working separately. Okay, so I wanted to tell you a little bit more. So often when I talk about what we do in the lab, I talk about a basic science question, and then I might go and talk about some of our disease models. But I wanted to talk a little bit about a developmental story that we have, because I think it's really interesting, and I want to share this with you. And I thought that, I know there are lots of people studying development here, so I thought this might be interesting. And I'm going to tell you a little bit about something we have been studying for several years. And these are called axonal, I'm going to talk to you about something called axonal swellings, okay? And so these have been studied for a long time. This guy, Baumann, he first identified them in 1918. Here is a Purkinje cell, and here's a swelling along its axon. I know you have to trust me on this, like it's really hard to see, but that's what he was showing. And he called these axonal swellings torpedoes, okay? So he was in the Netherlands. It was 1918, and 1918, the Great War was on. He clearly had things on his mind, let's say. And so he called these things torpedoes. This was, I should mention, this is post-mortem tissue from a person with schizophrenia. So these axonal swellings, first time they've been seen, what are they doing? He wasn't sure, but clearly by calling them something like torpedoes, it sounds maybe bad, right? And I'm not going to go through all the evidence, but these are seen in a lot of different diseases, and there's been this general idea. This is, I would say, the working hypothesis that we came in with, is that Purkinje cell axons, through perhaps some sort of insult, these axonal swellings form, and then that leads to worsening and maybe even eventual cell death, okay? That's a kind of neurodegenerative model of axonal swellings. And as I say, they've really been seen in a lot of post-mortem tissue from a lot of different diseases. So there's good reasons for thinking this. However, People hadn't really looked at what they might be doing functionally. So, two things. So everything I've told you about up until now was kind of focused on aging, disease, neurodegeneration, you know, but I told you I was going to tell you a developmental story. So this is where the developmental stuff comes in. So this is a paper we published a few years ago showing that we see axonal swellings in development. And we're not the first group that did this. It actually, Richard Hawkes from University of Calgary was one of the first people to show this in developing rat, and we're showing it here in developing mouse. And so you can see that at P7, we don't see it, so postnatal day 7, so mice take maybe about, you know, by about P21, the cerebellar circuit in the cortex is, you know, getting more mature. So you can see these don't look really like Purkinje cells I've shown you before. They're very juvenile. But by here, P30, these are pretty mature Purkinje cells. And there are still some axonal swellings, but we see a big peak of them in the second postnatal week of development, around P11. And that's just showing you this. So we see very few, we see this big peak, and then we still see some in adulthood, which we thought was kind of interesting. So swellings on axons, right? What could they be doing? What do swellings do to axons? We are interested in the swellings in a number of different ways, but we decided since they're seen in development, let's look to see what they're doing in development. For one reason, they're just easier to study in development than looking in neurodegenerative diseases when they're really, really old. Okay, so what do swellings do? Well, first of all, what do axons do, right? That's important. So I would argue axons do lots of things, but the basic thing that an axon does is carry information across space, right? In the form of an action potential. So people have modeled what swellings might do to an action potential, and they've come up with some ideas. You know, this is just a cartoon, but like maybe it reduces the velocity, reduces the speed of the action potential. Or maybe, okay, it's already kind of slow here, and then once it gets to the swelling, it just kind of fades on out. That would be an increase in action potential failure along the axon. So we, being electrophysiologists, decided we could look at this actually and try to see if any of these models were true. Okay, and when I say we, I mean a former graduate student who graduated and has left the lab. He's now working in biotech, Dr. Danek Leng-Ouellette. And so what Danek did, when I say we, I mean Danek, he was able to do dual recording. So one recording from the cell soma and one recording from the axon at a different location. Either an axon, which we're going to call control axons, or axons with swellings. And you might be wondering, why am I calling it a torpedo in some cases and a swelling in another case? We don't actually know if, you know, a swelling is the same as a torpedo, if the developmental things we're seeing are the same as the things that are seen in disease. So we're just being very agnostic and calling it a swelling. But you can see there are very clear swellings on these axons. Okay, so when he does this, what do we see? So here I'm showing you just an example, recording from the soma and a recording from the axon, soma and the axon. I think I first told you about the propagation, like that it could go more slowly. So we looked at the timing and between the peak of the somatic spike and the peak of the axonal spike, and there's no significant difference there. So it looks maybe a little bit shorter, but that's not significant. So it's not this idea that it's making the action potentials propagate much more slowly. Doesn't seem to hold true. So we say no, that's wrong. What about this idea of failures, that they actually go and then they fail when they get to this big swelling in the axon? Okay, so what I'm showing you here is an action potential in the soma, and what do we see here in the control axon? That's a failure. So I know it can be kind of hard to understand what a failure would look like, but in this case here we see an action potential, soma, axon, soma, axon, soma, It's not there in the axon. So that action potential did not propagate down the axon. That was not conveyed to the downstream targets in the CN. Okay, so that's just so you understand what an action potential failure looks like in an axon. So then we do the same thing in the axons with swellings. Where's my example of a failure? It is not there because this is actually what we see. Axonal failure rate is low in control axons. Most of the action potentials get down the axon. That's what axons are supposed to do, but not 100%. Sometimes there are failures. In our control axons, we sometimes see failures. In our axons with swellings, we almost never saw failures. They just seem to propagate action potentials even better. So we don't think that there's an increase in action potential failure. In fact, we think the opposite is true. We think swellings reduce failures. Okay, so if swellings seem to counteract failures, we have this idea that maybe failures are what cause swellings. So does spike failure lead to swelling formation? So we actually did a little trick. You guys are probably familiar with TTX, which blocks voltage-dependent sodium channels. And usually when you see an experiment that does this, people use what we would call a saturating dose to block all the action potentials, all the voltage-dependent sodium channels. So we use subsaturating levels of TTX to just block some. And what I'm showing you here is a recording from a soma and an axon, and then we wash in what we're calling low TTX, a subsaturating dose of TTX, and you can see, well, there's a lot fewer action potentials, but in the axon, there's basically none left. And this is some summary data, and you can see that in the soma, we have more action potentials than we do in the axon. This is probably just because of the concentration of voltage-dependent sodium channels in the site of initiation versus the nodes of Ranvier along the axon. So we kind of use this as a trick to mimic axonal failure, kind of an extreme case of axonal failure, because basically the axons are failing here. So when we do this to an axon, what we see is that within a couple of hours, we can make these swellings form on the axon. And here's a little time-lapse image showing just that. So it's a real kind of gradual process. If we use saturating levels of TTX, high TTX, we don't see that. So this isn't just something weird about activating voltage-dependent sodium channels. We really think that it's about failures along the axon. Okay, and this is just summary data showing you that this low TTX allows us to see the formation of new axonal swellings. We know that these swellings require calcium. If we block calcium in the extracellular solution, we can completely block this, even with low TTX. So it's not just some weird thing with TTX. It really seems to be a kind of functional thing that requires this, whatever is happening here, it requires calcium. You might be thinking, okay, this is interesting, but these swellings are just some little things on axons. You're seeing changes in action potential propagation, but does that really matter? Well, we wondered those questions too. And so we looked, we did the following. One of my undergrad students had actually told me that sometimes we saw lots of swellings, sometimes we saw few swellings on an animal-to-animal basis. And that made us decide to look at look at a question of like whether there's variability from animal to animal and if that correlates with anything. So we looked in this anterior part of the cerebellum that I've already told you about that's really important for motor control. And we looked at a behavior we called rotarod, which is allowing us to look at motor coordination between limbs. And so mice get better and better when they do this. On day 1, they're kind of not so great. By day 7, they're doing a lot better. Most of them are learning. And you can see there's a lot of variability. Most of them learn, but not everyone. These are individual animals. So we're basically using the natural variability across animals And then we looked at the variability in the amount of axonal swellings that we see in the same brain region. And it's big, right? Some animals have a lot of swellings, like 60% of Purkinje cells seem to have swellings, some have few. Is there a relationship between that? So we looked at the amount of learning and we looked at the percentage of swellings, and we're able to show that there's a correlation. So low learners had few swellings, and high learners tended to have more swellings. Obviously, this isn't the only thing that influences the animal's capacity to learn, but it really suggests that these swellings matter and that they actually are helping the axon function better, and that makes the cerebellum work better. So that working hypothesis I told you about, we think it's just wrong. So we have a new model where we think that axonal swellings are a kind of homeostatic mechanism to detect axonal failures. So when there's axonal failures, you have the formation of a swelling. And that actually improves the function of the axon and, of course, makes the Purkinje cell very happy as well. So I'm going to leave it there. We're continuing to work on this. Most of what I told you about this was published, but I have a new graduate student, Bruna Suarez de Souza, and she's been looking at axonal swellings in development. I have a postdoc, Amy Smith-Diak, who's looking at axonal swellings now also in an animal model of disease where they're prevalent. I wanted to just acknowledge Kim Groover, who did most of the work on the mapping of the inputs, and of course Eviatar Fields and also Danek Langoulet, who did most of the work on the axonal swellings, and everyone else in my lab, and thank you for your attention. Any questions? Yes? Should we do students first?
Sure. 2 questions.
Great.
First, thank you for your presentation. It's really informative.
Thank you.
Do you know if the swellings are still myelinated or do you lose myelination in their region? And they kind of seem to be the same distance from the axon L.
Yeah.
I guess do you observe them somewhere in the middle or do they vary in that location?
Those are great questions. So I didn't tell you because I went through it pretty quickly, but as far as we can tell, these swellings are myelinated. So this is, we're really unsure why they're arising at this time during development. And one of our ideas has been that, it's critical that the Purkinje cell axons work well. And so maybe, we still think that myelination might be involved, but they are under myelin. And so we've done some EM and we've been able to look. The myelin isn't like thinner. It's basically the same thickness as the rest of, we don't know for sure that they're all Purkinje cell axons, but just like the larger axons are probably Purkinje cell axons, and they have the same kind of thickness. So so we don't know about that. As far as their location, that's an excellent observation. And we haven't really understood yet why they are where they are. It is not the node of Ranvier. That's something that several people have asked me, and we have actually done some labeling for nodes of Ranvier. And they are close to nodes of Ranvier, like within, some of them are, close, some of them are a little farther away. So it doesn't look like there's a strict relationship, but yes, they're kind of located, maybe 50 to 80 microns from the Purkinje cell layer, and they do tend to cluster. So we have really wondered about that. But you also see axonal swellings, so once all those axons go through the granule cell layer, they kind of bundle into this white matter. It's going to be really hard to actually record from them there because there's just so many of them. But there are definitely swellings in there too. So we don't think that there's only necessarily just one swelling on the axon. There might be further down more. I think that answers your question. Yeah, thanks. Anyone else? Yes?
Thank you for the presentation. So you mentioned there were a lot of cells that were unresponsive when you were recording.
Yes.
So does that mean the slices are cutting a lot of inputs? And
Yes.
Is that relevant?
Yeah, definitely. So that's a really good question. Yeah. So basically, some of the cells don't respond, but we know that they were patched well because if you stimulate right over the cell, you can get a synaptic response. So you can directly activate. So yeah, that really suggests that those axons are probably coming in, like we slice in this parasagittal plane, and then there's probably coming in just out of that plane, and so they're cut before they kind of, we're not able to activate them in the lobule. So we're recording, so there's actually 3 different cerebellar nuclei. They're all kind of next to each other, and we're recording from the fastigial, which should be the one that's sort of the most direct input from sort of straight up and down, but then it kind of fans out, and we definitely think that there's like more coming, like we wouldn't be able to make a slice preparation that would preserve them if we were looking further out, and even here we probably sometimes are missing some. So if that's true and we have axons cut, it would suggest that, some of the cells that we think are 1 zone or 2 zone inputs could be 3 or 4 zone inputs. So if anything, like we find an overrepresentation of these 4 zone cells than we expect, but our data would be consistent with we're underestimating how many of those cells are there actually. So I do think it's important, but this is kind
Thank you, Alanna. I was going to ask a couple of questions as well.
Yeah.
I noticed there was some variation in the input amplitude you were recording when you get record from the CN cell, and I wasn't sure if that was just variability from prep to prep, or actually is that variation correlated with the different zones?
Right, yeah. So I did, let's see, maybe I didn't go through it. Yeah, actually, this is looking at the zone, so I had it as a supplemental, yeah. So basically, if we look at the zones like 1, 2, 3, 4, and we look at the amplitudes, there's no significant difference. I mean, it's kind of a trend, but if we kind of sum them up, like if we look at, yes, they sort of summate, so suggesting that when you get, like the 1 zone cells tend to, I mean, it's just a kind of trend, but they tend to be a little smaller than the 3, 4, or 5 if we look at like the sum of the total input. So there doesn't seem to be, what we sort of wondered was, would we be hitting kind of cells that was, predominantly getting input from 1 lobule or something like that? That doesn't seem to be true, but there may be, with a lot of this data, we're limited by, we have so many different patterns that it's like really hard to, like, it's hard to get large enough sample sizes to really see. So there could be differences that we just haven't detected yet. Yeah.
And my second question was, do the swellings go away later in development?
Yeah, great question. So, yeah, like they do. I showed you that they kind of go up and then down. So they definitely go away because Purkinje cells, like there is sort of programmed Purkinje cell death that happens earlier. All the Purkinje cells with the swellings are going to persist throughout the rest of the lifespan. So yeah, they definitely do. We've never seen them go away, and we have done a lot of time-lapse imaging, like a lot. So we don't have the conditions right to see them go away, and we would love to understand what the conditions are that make them go away, and I don't know why we haven't kind of occasionally seen one go away, but as far as we can tell, we haven't. But I think for that, maybe the time scale is just longer. So we're doing this in slice, the time-lapse imaging. We would like to do time-lapse imaging in vivo, and maybe it's just like in slice, it's going to take way too long because things are kind of probably a little, my guess would be faster in vivo than we would see in a slice. Yeah.
I was just sort of thinking about differentiating between torpedoes and swelling.
Oh, yeah. You mean like the
Torpedoes really are something because of that one example
Yeah.
of a disease
Yeah.
that are not typical brain.
Yeah, and that they're kind of end of life. Yeah, so yeah, because that's all like post-mortem human tissue. So it's really like end, yeah. So I can tell you that Amy, my postdoc, has been looking at these in our mouse model of RSACs, which have torpedoes form very young. I think we can call them torpedoes. And they do look, it does look like they're kind of 2 populations. We think there may be sort of the diseased ones are maybe located a little closer to the soma, so they seem to be a different depth and also a little bit smaller. So it may be, we don't have a good understanding of exactly what's different, but that there could be differences. And so they're like superficially look like the same structure, but they're not. But we don't know yet. Yeah. Yes. Yes. Sorry.
No,
my pleasure.
I'm really impressed by the relationship you see between these swells wondering if maybe one function or one way of thinking about these effect of these swellings, which is that they never drop out in action potential. I mean, that's
Yeah, that seems to be
so
true.
Can you
Yeah.
think of these random losses of action potentials as a source of noise?
Yeah.
So if you have a neuron that has a swelling, it has less noise and better signal-noise ratio or something.
Yeah, yeah, I think so. Yeah. And I mean, it is kind of interesting because you could kind of imagine that a little bit of noise might be good for the circuit, but certainly it does seem that the animals that have more swelling seem to do better. But then what's underlying that? Like why some animals have more swellings and some don't? It's kind of like we're kicking the can, right? Because we still don't, is it like some animals have a better predisposition and therefore, yeah, I don't know. But I mean, that's, I guess, always a question. Why do some animals learn things better and others don't? Yeah. Yes.
Do you have any hypotheses so far that might provide a mechanistic link between the swelling and the lack of action potential properties?
That's a great question. So the question is sort of like, so I'll say my hand wavy thing is like, you know, we all learned about axons with the giant axon, which was giant, we think, because of it's involved in escape response, right? It's like action. There is some precedence that axons get large to become more effective. However, I would say a large axon, uniformly large axon, is very different than a swelling. So I don't really, we're working on it. We're actually collaborating with a postdoc in Eve Marder's lab, Yun Liang, who is really interested in action potential, in axons, and we're really hoping that we can get to some sort of understanding about this and like what exactly it's doing. So we've done a little bit of EM, as I said, I didn't have time to show it. There's nothing different about like these structures, like the density of organelles, like mitochondria and things like that. It's sort of the same as the other axons. So we can't see anything that's making them different. So I don't know. I mean, I do wonder if it's just, you know, it could be that there's something very specialized, localized there. We know that IP3 receptors seem to be expressed there, so there would be endoplasmic reticulum. Or is it like a biophysical thing, like of having a swelling that makes this axon function better? I don't really have a good understanding of that yet, and I think it's such a big open question. But I think modeling is probably going to be the way to address it. Yeah. Thanks. Yes.
I was really impressed by a lot of these experiments, and I was just kind of wondering what nucleus did you record from, and also I'm assuming, I think it's maybe the medial because of the breath, but
Yeah, the vestigial, right?
Yeah.
Yeah, which is also, the
I'm
medial.
wondering if you looked at any other nuclei,
Uh,
or also, do you think maybe some of these cells are a certain cell type in the DCN?
Great questions. So I'll definitely pass the feedback on to Kim because yes, it is really beautiful work. Great question. So yeah, we don't know. I mean, there is some evidence that different nuclei, I mean, that they have slightly different projection patterns and things like that. So it could be just this nucleus and that different nuclei are different. That's going to be hard to address with a slice prep. You'd have to really kind of, I don't know, yeah.
It seems hard enough as it is.
Exactly, yeah.
I'm just kind of-
Kim really wanted to repeat all this data but then at some point she was like, oh yeah, should I have a 5-year PhD or a 10-year PhD? So maybe not, because it's not like it gets so much faster as it goes on. But your other question, I'm just blanking. Oh, cell type. Yeah, great question. So had we been thinking, like there's so many things that Kim did with this. She actually would do this, record the location, she would image the cell, all these things that she did. I didn't show this. There's no morphological differences in the cells, the different classes. They look really, really similar. But like there are new papers showing that there are sort of expressional markers for different projection types. We didn't think of doing that. We saved all our slices. We post hoc stained for them, and it just did not work out. So we were not able to identify the cell types. We would have liked to have. Yeah, the Sacha-Delok different types of projection neurons. And it's just like, it was just already super complicated, and it just didn't work out. So that was a real kind of blow. We felt really bad, but there's only so, it's like we've done so many things to this little scrap of tissue, and then, yeah, so we didn't know. But we'd love to know that, and I think maybe the way forward would be to get like markers for the different cell types so you target them rather than doing it post hoc. Yeah. For the next person who comes to the lab and wants to work on this question. Yeah, yes.
Those CN cells, the activity of them seem very, very regular.
Yeah.
And maybe it's just the example, but if you move around and record from different CN cells, do you get high, is it very tight, or is there different zones of activity?
Good question. So I would say they're pretty regular in a slice, right? So in a slice, when presumably most of the input has been cut off, so they're not being driven by input, because these cells do, like I'm talking about, okay, the Purkinje cell input is one of the main inputs that they get, but they do also get excitatory inputs from climbing fibers and mossy fibers. They get collaterals that come in as well, so they do have excitatory drive as well. So to be honest, what's driving their activity is more complex, and certainly in vivo, the patterns are more complex than you see. But I actually think this is a strength of the slice preparation, because it's kind of giving us this baseline, like tonic activity that everything else is riding on top of. Yeah.